All right, welcome back after lunch. My promise to you is this is probably one of the top 2 genomics talks on vanilla during the conference. So you're in for a treat. All right, so thanks everyone. We've made some good progress. I believe I've got Josh doing the admin on the other side, so I can take a break and share with you a little bit of my passion for vanilla, which is the genetic improvement of the species. All right, so just a little bit about my background. So I am hired to create new cultivars of tropical crops, and vanilla is just one of those. I only work on crops that I think are delicious, which actually means I'm going to be eliminating papaya because apparently it doesn't agree with me, but everything else I work on.
Doesn't make it less delicious.
To everyone else. Before here, I came from PepsiCo, so this is my claim to understanding what it's like in the private sector, but also understanding what it's like here in academia. And really, I'm at home either way, which also makes it nice for all of our industry partners for me to understand what it's like for them to meet their milestones as well as us to deliver on. So I used to take more time to show this slide, but basically the general overview and the whole reason civilization exists, and not just because I'm involved with agriculture, is that agriculture exists, therefore the rest of us can have other jobs, okay? So if you look at wild crops, you have things like this tahitensis, small grains, hard as a rock, and they had to be specifically prepared to release their nutritional content versus today's modern improved cultivars, which are these high yielding sweet corns or field corn, which is what we use to feed our cows. So you look at something like a wild apple, these little teeny things that might look like cherries that taste absolutely terrible versus today's modern apple, not the Red Delicious, but any apple except for the Red Delicious apple. Things like seeds and bananas. I bet no one here had banana. Their last banana had seeds in it, right? This is because of domestication. Look at this wild carrot shown here. There's not a lot of food there, right? You're not going to sustain a population of people off of these wild crops versus today's crop, highly nutritious, highly abundant, disease resistant, and this is how we're able to enable society. And yet we look at vanilla, and vanilla is a wild species. It is the tahitensis, it's the wild apple, it's the seedy banana. So while vanilla is already great, when I'm looking at it, I see a wild species and we think, where do we want to go next, right? And if we can turn this into that, we can do a lot with this as well. Okay, so it's important to know how we got here. Otherwise, you've got no perspective, right? So this is the first botanical notice of vanilla so this is in the literature, right? So we're going back to 1605. You can practice your Latin, it's in here somewhere. You've heard people say vanilla planifolia Andrews. This is why they do that, because Andrews was the first one to draw. Here's the vanilla vine. I personally don't agree with that because you've turned a Latin binomial into a trinomial, and that's the whole point of genus and species. You have 2 names. This one has 3. I like Andrews, but I'm going to call it vanilla planifolia. All right, so you've probably seen variations of this slide before. So where does our vanilla come from, right? So we know it originates in what we say Mesoamerica, so southern North America through Central America. It started this migration route, maybe as early as the 16 to 1700s, right? But we know that the Aztecs, where they valued the vanilla bean, the vanilla spice, and then it was actually cultivated, we think, for the first time by the Totonac people in Mesoamerica, right? It started spreading in the 1700s to Europe, but it wasn't until much later after that that they were able to keep the plant alive. So you take a tropical plant, you put it in England, and it dies of depression, right? And then they figured out how to keep it happy. We know that vanilla planifolia has been in Florida since at least 1910, and I'm actually moving that number back, and I'll show you some things that I think are interesting. It also had a trans-Pacific route through Hawaii to the Philippines, and then through the islands of the South Pacific, Indonesia, through here. And then, of course, bouncing to Madagascar, probably around 1840 or so from Reunion. And then we have artificial pollination. So recognizing the fact that you could have a vanilla plant and have beautiful flowers, but never get a bean outside its native range until people like Charles Morin and Edmund Albius figured out how to pollinate these flowers manually, right? So 1800s. So vanilla has come a long way, but we also haven't been working with it for very long. So vanilla in Hawaii, I'm going back into some relatively old literature here. We know it was there at least by 1903. Some nice pictures, and they were working on a commercial industry in Hawaii back in the early 1900s. It's interesting to read that they had some of the same problems that we still have today with viruses, disease, and how to cure the beans, how to keep the plants healthy. So you'll see that we have these cyclical patterns. A plant will go and get established, then it will get lost, and then reestablished, just like what happened in Puerto Rico. So Puerto Rico actually had a vanilla breeding program until the mid-1900s. And so you'll see these pictures, and maybe you'll mistake them for what happens in Madagascar. Not this one, but Tudor trees, a slat house. We've got concrete pads where they're curing the vanilla beans. But what we think happened is that in response to industrialization and because of fusarium problems in Puerto Rico, the industry collapsed. So now you only have feral populations. And with some of the work that's being done in recent years, they're trying to bring up that industry again. And then in Florida, so I've been digging into these old references, pre-1900 now. So these are USDA records. It wasn't called USDA records back then, but telling them that they were importing plants from France, from Mexico, from the Bahamas, most likely not vanilla planifolia, and then again from Mexico. These were some of the very first introductions into Florida, and we think that some of them actually still exist in some of our protected lands today. And the guy with the red circle around his head, that is, what's a swingle in that 1899. So this is what people like me used to look like. And I'm glad that our dress has evolved. Usually, I wear a t-shirt. Can you imagine wearing the tie and you're out in the jungles looking for plants? And I'm maybe not a very diverse crowd either, but so in vanilla, we in Florida, so those plants came to Florida starting in around 1900, right? And then these records indicate that there were these non-persistent I would call those feral plants. So they were one time protected and cultivated, and then someone walked away from them, and the plants were just left to their own. And this is what it looks like in this first slide here, or this first picture here. This is in the Deering Estate. This is protected land. It's an archaeological preservation site just up towards Miami. And there's vanilla in there. This might be some of those very first plants because we know that there were these native settlers in this area, and this is land that has been, you haven't been able to develop, right? And so they've just got a couple of plants there. Of course, we took those plants and we have them here as well. This picture with the hand and the bean, this is a naturally pollinated vanilla bean. One of the biologists was out there one day. She was like, oh, look at this. Took a picture. And then a year later, I was talking about, oh yeah, I've got a picture of this plant. I'm like, you got a picture of a bean. And this one on the far right, these are naturally pollinated vanilla beans from Pine Island, the actual island, right? Where some of you are from. We don't know what's pollinating the flowers, but we're going to find out and see if that's something that we can rear to affect pollination in Florida. A little bit more perspective. So as late as 1944, Mexican vanilla beans were still the definition of quality. I have gaps here. If anyone has more information on historical trends pre-FAO, I would love to see that information. Where the beans are coming from, how much they're worth. We all talk about Madagascar, but we have to recognize that Madagascar had to start somewhere. And before Madagascar was someone else. It was Mexico, right? And at the same time, we appreciate what Madagascar has done, but that doesn't mean they're the only player now and forevermore, okay? Okay, so now I want to talk about some of the fun work we've been doing with our vanilla. So we know that vanilla planifolia has the center of cultivation in Mesoamerica, Central America into South America, okay? But we also read in the literature that there's not a lot of diversity out there. But if you take a plant and you clonally propagate it for 100 years, you're not going to develop a lot of genetic diversity. So we go to Madagascar, the real diversity we're going to find are only those few remnants of the breeding program they had until the 1960s, okay? So what we want to do is go back to these areas that should have wild populations and diverse material. Why? So in plants, diversity is where we get the genetic tools to solve our problems. We want to go from that wild plant to something better by combining the best genes from mommies and daddies and making superstar children, okay? And then this picture with the on the far right, we've been reaching out to people in a number of different countries to characterize germplasm and understand vanilla as a whole. And these are the countries where we have people that have been willing to work with us, sending us leaf material, sending us plants, and then we share the information back. We publish together. I think it's a nice open model that's working well for people. And again, it's helping us to not be so narrowly focused on our view of vanilla, but taking a wider perspective. A little bit more historically. So I'm going to talk about DNA work, and we can think about this like a fingerprint, right? So your fingerprints are unique to you. So if you commit a crime, we know who it was. Especially don't do that on campus. There's paperwork we have to do. But for vanilla plant, instead of using a fingerprint, we're doing that with the DNA. So we sequence a small piece of DNA, and then I can tell you what species you are, right? So here's a stretch of DNA, which you might have seen in your biology classes a while ago. And then we have these single nucleotide polymorphisms, or a single change in that DNA sequence. So one person has an A and someone else has a C. We can use that, and then we can track that difference to help us identify who is who and who's not who, right? You do something like this, you spend $5 and you get maybe 5 data points, okay? With those 5 data points, you make a family tree. So you can take a bunch of unknown samples, I check it against my database, and I say, you're in the blue family, you're vanilla planifolia. Oh, look, you've got this other, SNP, you're in the Pompona family, so we can identify a species. But the limitations are things like hybrids, like Tahitensis, you can't tell it apart from its maternal parent, planifolia. And you also can't differentiate other hybrids. So if you've got a planifolia Pompona hybrid and you do this test, you can only tell who the mom was. You don't know that the dad was there, even though it's contributing about half of its DNA. Okay. So we took that to the next level, and instead of using 5 data points, we're using about 15,000 per progeny. And what that does is increases our resolution, our ability to differentiate different types of vanilla. And we create a massive family tree. When we did this, there were a number of things that stood out, and I'm going to explain. Okay, so what's going on here? You've got a family tree, and the closer you are, the more related you are. The farther you are, the less related you are. Okay, so we've got a distance component. Then you've got these colored blocks at the bottom. The colored blocks are based on patterns of DNA. So if you sequence a bunch of common planifolia plants, it recognizes in the algorithms, we have this block of very similar DNA patterns, and it gives you a color, pink. Okay, nice, easy to see. So your typical planifolia is going to be pink in this figure. Some of the things that popped out on the far right, this is vanilla tahitensis. It took us a long time to get verified vanilla tahitensis because everything we had purchased came back as planifolia or phantha or planifolia phantha hybrids. And now we can hold people accountable to that because we can tell the difference between them. So we found tahitensis using our DNA fingerprint, but then we found 3 different kinds of planifolia, right? So you have your common planifolia, which is what we call type 1. Now we have 2 other types. Type number 2 is very similar to tahitensis, so much so that we think if you were to take out the odorata from tahitensis, what you'd be left with is type 2 planifolia. So that might actually be the dad of vanilla tahitensis, and yet we don't see that in cultivation widely. Does it have any unique traits? Does it contribute different flavor profiles or something to tahitensis? Now we can actually go and target those plants and ask those questions. We have a vanilla planifolia type 3, same thing. It's genetically distinct. We don't know what that means yet, but we're certainly going to go that way. We have blocks shown here in this dark blue. This is vanilla pompona, palmarum, insignis, odorata, etc. And then if you go far over to the right, these are planifolia pompona hybrids. We find these all over the place. Costa Rica, we find them from botanical gardens. We have them in our collection. But now we're able to accurately identify which ones are hybrids and which ones aren't. All right. If you spend 2 years generating this figure, it's a lot more beautiful than other people appreciate. All right, so I took a snippet of just the kind that you guys care about. What you guys really want is tahitensis and planifolia because you want to stay in that legal definition. And we're able to take that genetic information and start separating things out. So one of the things we found was a heterozygosity level. And this is actually contributing to what makes a plant different from another. So you've heard stories about inbreeding in Europe, right? And with the aristocracy centuries ago. And that led to a number of genetic problems in the royal families, right? Because of inbreeding. But we're finding some vanilla plants that are much, much more inbred than others. What does that mean? I don't know. How did it happen? Again, we don't know. But it's enabling us to say this type is not the same as your common vanilla because of this level of heterozygosity. And then these are PCA plots. They're separate. They're taking all that information, finding general patterns, and then separating things that are different. And we can find all these different species that are splitting out, but then we're also finding these different groups of planifolias that are splitting out, and then as well from tahitensis. And then if we look at just the planifolia, tahitensis, and what we call Soto-Orentii, we find these big groups. I've had people ask me about Soto-Orentii, whether it's a distinct species. That's a botanical question, but as far as I'm concerned, it's 100% planifolia. It will meet the, because you can't, I mean, you're not gonna be able to dig into this data and say, no, there's justification, this is a unique species. And because of that, it's an interesting type. We're gonna go look at it for interesting characteristics as well. All right. We like pretty pictures of flowers. This is a reverse tahitensis. So the tahitensis that you commonly know has planifolia as the mommy and odorata as the daddy. This one swapped it up. So its daddy is odorata and its mommy is planifolia. Do we care? Well, in roses, if your maternal parent has a specific gene, then your progeny smell like a rose. If you swap it and you use that same smelly plant as the dad, you lose the rose scent. So we start asking, does your mom really matter? Well, I mean, of course, in humans it does, but in plants it doesn't matter, right? And now we can say, okay, now we need to go and we need to get this plant. One of the benefits of our approach is that we can sample hundreds, if not thousands of plants, and then pull out the ones that are unique and special. So instead of focusing on 1,000 plants and doing all of the aroma analysis and all this stuff, we can pick out my 10 favorite and we can focus on those. Now, this one doesn't exist in a living collection. It's in the jungles of Colombia somewhere. And so instead of telling them to go and say, go get everything you sent to me and collect it and preserve it for all time, here's your top 3. Go get those ones, maintain them, and let's see what they're doing. Like I said, we found that Soto-Orentii is planifolia, as far as I'm concerned. We found some interesting differences in odorata. Again, for those of you who like the tahitensis profile, now we've got 2 different options for daddies that might help us recreate new and better tahitensis in the future. And that's shown here just by the differences in the flowers. I talked a little bit about conservation. Of course, we know that vanilla is endangered, as I know people need to make a living, but we can find a balance at the same time, I think. So this publication just came out from Crop Trust, giving us some general guidelines, taking surveys. Some of you contributed to this information on what we can do to preserve some of these jewels of vanilla types before they're lost forever. And of course, the strategy we're taking, not that I want to encourage deforestation, but we can say for sure this is unique. You should protect these plants with more effort than 100% of everything, right? Because we can't just justify saving everything. And then with the collection we have now, people have sent me plants. We maintain them here at the University of Florida, thanks to the generosity of University of Florida. Last year, we had a number of hurricanes, like we like to do for fun in Florida, but 2 of them hit Nicaragua, one right after the other. And we had some grower cooperators we were working with, and they lost everything except for the plants that are in our collection. So now we can send them back plants, and then they can build up their material again. So this is the argument for creating these living collections off-site. And it would be great if I could do the same thing with my rare material. Spending 5 years and tens of thousands of dollars creating this, to think that one hurricane can wipe it out, that's a lot of risk, right? But for something like this, one country gets their stuff wiped out, now we can help reintroduce it, right? So we're de-risking this process. And then this is a picture of some of the different plants in the collection, which we'll actually do in person a little bit later. All right, vanilla genome and how we're using that to interrogate vanillin biosynthesis. So this was a major publication, a major research effort, public-private investment here to help us to read the genome of vanilla from start to finish. For you guys, it probably doesn't make a big difference whether we can actually do that. I mean, it does make a very pretty picture. I mean, look at that, all the colors and things. But those are the different chromosomes. We can sequence the mommy's chromosomes. We can sequence the daddy's chromosome. But now I use that as a tool, a very, very powerful tool that makes everything I do faster and cheaper. So we say, what can we use the genome as our new tool to understand how vanillin is made in the plant, right? We want more vanillin. And so we say, can the genome help us? Here's a biochemical pathway. I know most of us think that they're ugly, but it has its own kind of beauty, the arrows and things. There's been a number of papers that have been putting forth different models for how vanillin is actually biochemically synthesized in the plant. So one group says it happens way up here and it requires 2 different enzymes. Another group says, nope, as long as you've got one and it works down here, it happens. And it's stealing metabolites from lignin biosynthesis in the plant. So we can use the genome and we can look for all of these genes shown in this blocky figure here. And we're looking for a guilt by association analysis. So we take that gene and we look at different tissues of the plant and we say which gene is turned on in the right place at the right time. And just like you would do for forensics or a detective story in humans, if you've got a good alibi, we're going to get rid of you. And if you don't have a good alibi, you're probably guilty. And we went through the 45,000 genes in vanilla, and it turns out we have one favorite candidate. And it's not only just one, it's one flavor of one candidate. So this gene is present at the right place and at the right time. And it's not just that one gene, it's that one version of that one gene. So now we've got a hypothesis and we can screen hundreds of plants and say who has this and who doesn't. Does someone have 2 copies or 1? Do some have 0 copies? And that's why we have non-aromatic vanilla species. So again, it's about taking this absurdly large number of possibilities, bringing it down to something we can test very easily in the lab. And the result might be dry vanillin content of 8%, just because it's naturally got more capability to do that, right? All right, and vanilla breeding, which is the reason that most of you came to this conference, because you love vanilla breeding. So everyone says, oh, can we grow vanilla? Can you even do this in Florida profitably? Okay, so here's an estimate of the gross return, so gross, not taking into account investment, for vanilla in Southern Florida compared to some of our other major crops. So you look at something like strawberries and passion fruit and bell peppers. These are some of the highest grossing per acre crops in Southern Florida. Vanilla has a good shot, right? Much, much higher than even oranges, which is something we're well known for, okay? And I can show you the background numbers. It's relatively simple and I would say relatively conservative, the numbers that we use. We're certainly not using those peak numbers from vanilla prices in the 2018, 2019. Of course, we work with a lot of growers. We have 17 growers here from South Florida. Understanding from them who they are, what their interests are, and what they're growing on. Out of the nearly 400 growers or interested parties that have responded to our surveys, we found about a quarter of them are interested in commercial production. And most of them have small acreage. You're talking 1 to 5 acres. And many of them are diversified. So they're growing mangoes and vanilla or passion fruit and something else, and they're just adding to it. And then where can we actually grow versus who's represented? In the figure of Florida here, these are counties of people that are interested in joining our network of vanilla enthusiasts. And this dashed red line is an approximation of where we think we can grow them. So with conventional plant breeding, historically, you've had, say, a mama plant, we'll call vanilla planifolia, that we all know smells and tastes great. And then we have vanilla pompona that might have something like disease resistance. What we want to do is take the best from both and put them into one. A lot of our tropical crops today are the result of pure chance. You happen to have one plant near another that were pollinated and that seedling grew, and that's how we got tahitensis. So by rolling the dice, every now and then, nature gives us something amazing that has a disease resistant and it tastes good. And it has a certain amount of space that it takes to accomplish that, a certain amount of time, and a certain amount of money. Using our DNA and genomic tools, it's much more calculated and measured so that we need less space. It takes less time because we're getting rid of more of the garbage, and it also takes less money. At the same time, instead of looking for just 2 traits, we have the potential to look for many, many traits. So we could grow out 10,000 seedlings, use our DNA fingerprint methods, and select the best tasting, the highest yielding, et cetera. This is what they're doing in so many other crops that we enjoy. Just hasn't gone to vanilla. What type of vanilla do we have in Florida? We see a lot of planifolia. We see a lot of pomona, That's this yellow flower, pretty diagnostic. It's vigorous, it's hardy, it's disease resistant. We also have a lot of sterile pomona in Florida. And so people will come to me, why does my plant not make beans? And if they're not pollinating, if they are pollinating, there's a lot of these sterile hybrids. So a grower can invest in these plants and never get a bean off of them. We also have 4 native species. Vanilla phantha, shown with this wide open cream colored flower. Vanilla mexicana with this beautiful white labellum. And then we have 2 leafless species, Vanilla barbellata and Doloniana. And I've got a picture of a bee here because we are actively trying to figure out the insect pollinator of our native vanilla orchids. So these beans shown here for phantha and here for mexicana, these are naturally pollinated. So no one was out there in the swamps pollinating these beans. So some insect was doing it. We're trying to figure out what it was. Maybe this one, probably not, but we're still looking. What are some of the things that the future vanilla plants could have? Of course, we want them to taste great, but we also want them to grow well. So we want a lot of bigger, strong plants. Wouldn't it be great if we could let the vine or the beans do like Tahitensis does, where they can ripen on the vine without splitting? You can go harvest at one time after they've all reached their peak physiological maturity. Of course, we want disease resistance, and we want to get away from pollination. As in, we don't want to do it ourselves. We want something else to do it for us. We talk a lot about uniformity, and where this came from is that we bought a number. We bought a number of Tahitensis plants, and they came back as planifolia. So I thought, wouldn't it be nice if we had a simple DNA tool we could use to identify right off the bat if it's the right thing or not? And so that's what we've developed. Specifically, we can differentiate planifolia, Tahitensis, and pomona. Odorata too, because it's part of Tahitensis. But it's even more impressive if you know anything about how DNA degrades. I can take a cured vanilla bean and I can tell you whether it's legit Tahitensis or not, right? And so sometimes you're buying a bulk of something, and I mean, those of us have been to a lot of these countries, they don't really care what they're giving you as long as you're giving them money. So now we can hold people accountable, at least on my side, especially as I'm getting plants and things. I'm not going to put more effort into something that we think is a Tahitensis when it's actually a pomona hybrid or something. And of course, and this is what the actual data looks like. We can go from a leaf to a result in 1 day. And so it's a rapid test. It costs a couple of cents. And then we can differentiate, put things in their proper pools, and then tell you what it is. As well as we're looking at our, we do aroma profiling here with our HPLC. So working with people that know a lot better than us, people like Daphne, to help us to understand how vanilla grown in Florida might taste different, at least be analytically different, and then we'll submit that to sensory evaluation to see what we've got going. For last year's harvest, we analyzed pomona beans from Pine Island, and they came back as an average of 3.5% vanillin by dry weight. So that was good. They're also very big beans, and that's what some of this data is showing. That's just 1 year though, so we'll have this year as our second year. Okay. I always get this question, so I'm going to hit it right now. Is this a GMO? And I have multiple ways of saying no. The short answer is no. The second answer is, can they be grown organically? Yes. That's another way of saying no, right? The long answer is that everything with DNA is genetically modified. Everything. You, me, the squash, the tree, everything is. The question itself is flawed because of marketing. And that's, again, we talk about terminology, and terminology should have value. For the most part, you can't purchase genetically engineered, which is a legitimate term, or GMO, which is a marketing term, or transgenic, which is also legitimate, cultivars for most of your crops. Okay, you go in the grocery store and you're not going to see those in the store. It doesn't work like that for most of our fresh products. Your genetically engineered crops are your corn, your soy, and it's not even sweet corn, it's field corn for feeding cattle. It's your soy, which is processed for oil. Maybe some of your eggplant, the BT eggplant, which they're growing in Bangladesh. And there's a transgenic papaya and a squash, which is virus resistant. Okay, that's about it. So when we think about what it means to be a GMO or genetically engineered, this is just straight up deceptive. No one is squishing their fruit with green goo using a syringe. That's not how it works. At the same time, when you talk about things like gene editing, we all love gene editing. No one's using tweezers to manipulate DNA. It's not possible. Okay. What we're doing is we are leveraging genetic diversity to tell us what's possible and creating new opportunities for growers. That's where we fall. All right, so I believe this is my last slide. Our next steps. We are still expanding our collection in genotyping. We're still looking for new partners, especially in areas through Central and South America. Of course, around Madagascar, we know there's a number of challenges trying to get material genotype there. This is one of my next focal areas as I really need to get to Papua New Guinea and some of those islands in the South Pacific, especially in February here where it gets real cold, like down into the 60s, and then bring material back. And what do we do? What do we offer? We can let you know what your plant material, how it relates to everything else in our database. Do you have something unique, something special? We can identify that and we can provide that back, as well as our living collection. Say you get a cyclone and it ruins your stuff. If we've got a living duplicate here, we can provide that back. Okay? And that's something we're offering as we share across these different countries. Of course, we're supporting domestic production. Part of that is bringing growers here, showing them how it's done, de-risking the process, developing new cultivars. Again, this takes a number of years, but honestly, if we would have started this 10 years ago, we would have some amazing stuff by now. So we're going to start 5 years ago, and then a couple of years, we'll have some amazing stuff as well. Creating resources, everything from genomics to genetics, to all the different tools we're developing, and of course for academia, so the information goes out. We're looking more towards our sensory panels to get a grounded reality check on our analytical data, and of course our conservation angle, thinking that we can use the various tools to identify the highest priority clones and keep those preserved so they're not lost forever. And with that, I'll thank you for your time. I think I did pretty well on time. Go ahead.